So, hello, good evening students, how are you? So, today we will, before starting my lecture, just confirm with a thumbs up that you can all hear me clearly. So, today we are going to start a new chapter. We have already started with classification, we have started with 5 kingdom classification, and we have seen 4 kingdoms we have already covered. Yes, good evening Pinky, Praveen, Anil, Khalil, good evening all of you. So let's start. As I can see that you all can hear me clearly. So we have already covered the kingdoms. So 4 kingdom is already covered. The last kingdom, animal kingdom, that will be covered in zoology part. Now, so today we are going to start a new chapter. And this is a very interesting chapter. You all must have been studying this chapter from your standard 6th, 7th, 8th, 9th, 10th. So all of you must be very much aware about this chapter, comfortable about this chapter. So it doesn't mean it is going to be very easy and nothing new you are going to learn. You will learn a lot of things, but yes, as a chapter, this you all must have been studying. So any guess which chapter I am talking about? So I am talking about the chapter cell. Now all of you must be having a smile on your face because this cell chapter we used to study from the beginning only. When you start studying about biology separately, you will study about cell. So basics of cell. So if I start with the basic question which always you study, what is cell? So this is a very popular question you all must have written several times. What is cell? So as written here, it is the fundamental unit of life. Cell is the fundamental unit of life. Along with it, I can use 2 more terms. It is the structural unit. It is the structural unit and it is also the functional unit. Functional unit. So can we say that in terms of complete definition, we will say cell is the fundamental, structural and functional unit of life. Cell is the fundamental, structural and functional unit of life. Now one question comes. Fundamental means what? It is the basic, it is the smallest unit of life, right? Any life cannot be smaller than a cell, right? The smallest living organism can be a single cell. A living organism cannot exist which is not a cell. Now, so that's why it is called fundamental. Why it is called structural unit? It is called structural unit because all living organism is made up of cell. Living organism is made up of cell. Whether it is me, you, plant, animal, unicellular, multicellular, algae, fungi, any living organism, all are made up of cell. That's why it is the structural unit. And why we call it functional unit? Because all the function of cell, all the function of body is performed ultimately by the cell only. For example, oxygen is transported in our blood. How oxygen is transported? It is the RBC who is transporting the oxygen. Similarly, signal goes from one point, let's say you are seeing a snake and you start running. So this vision and running action, the impulse is going through your cell. Which cell is it? Nerve cell. So all the function is performed. Digestion performed ultimately by cell, absorption by cell, respiration inside the cell. So all the function is performed ultimately by the cell. That is why it is also the functional unit of life. So now from here it is very much clear what is a cell. So definition part is clear. Now we are coming to the next part. As I said, these all part you all must have studied. Now we are getting into the new part. So here, first of all, in cell we study about a theory called the cell theory. Now question is, what is cell theory? What is cell theory? And why we are calling it theory? Because this cell theory will be applicable, it will be applicable to all living organism. This theory is applicable to all living organism. That's why it is called cell theory. So what is the main point of cell theory? So the main point of cell theory is all living organism is made up of one or more cell, and I can also add one more thing, and product of cell. Product of cell. So all living organism is made up of one or more cell. If it is one cell, then it is unicellular, and if it is many cell, then it is multicellular. Organism can be of two types, unicellular organism and multicellular organism. Now if I say unicellular organism, so they perform all the function, perform all the life function, all the life function. For example, you all are appearing for NEET, right? So all of you have to study all the subjects. Now let's say if I say that no, a group of 4 students will give NEET together. So what you will do? You will divide 4 subjects into 4 students. One will cover physics, one will cover chemistry, one will cover botany, other will cover zoology. So it is what? This is the function of multicellular. In our body, we are multicellular. So cell has distributed the work, right? But in unicellular organism, all the function is performed only by the single cell. Just like you have to study all the subject. You cannot say that I will only cover physics. So you are functioning as a unicellular organism. So in unicellular organism, all the life function is performed by the single cell. But in multicellular organism, there is a division of labor, division of function. Then the second point says that cell is the basic unit of structure and function. As I said just, structure because it is made up of cell, and function because life function is performed by the cell. Then the last point, all cell arise from pre-existing cell. All cell arise from pre-existing cell. How does a new cell form? All living organism is made up of cell. But how new cells are formed? So new cell will come from the pre-existing cell through the process of cell division. So these are the 3 points you have to remember about the cell theory. Now coming to the next question, who gives cell theory? So cell theory was given by, see, these 2 scientists, Sladen and Schwann. Sladen and Schwann has given the cell theory, but it was incomplete and it was completed by Rudolf Virchow. So ultimately, they 3 scientists, Sladen, Schwann and third, Rudolf Virchow. So from here, if I ask you who gives cell theory, so you will say Sladen, Schwann and Rudolf Virchow has given cell theory. But if I ask you who completed cell theory, so Rudolf Virchow completed cell theory. What was the limitation of cell theory given by Sladen and Schwann? So Sladen and Schwann has just given these 2 points, right? This point was given by Rudolf Virchow. So that's why we say that Rudolf Virchow completed the cell theory. Completed the cell theory. This point clear? So now, but if we talk about who discovered cell, so this all of you must be knowing, Robert Hooke discovered cell, but he discovered dead cell. The cell discovered by Robert Hooke is the dead cell. But who discovered the first living cell? So first living cell was discovered by Leeuwenhoek. He discovered the first living cell. So if question is asked without mentioning living or dead, who discovered the cell? So you will say Robert Hooke. But if question is who discovered living cell, then you will say it is the Leeuwenhoek. Who gives cell theory? Sladen, Schwann and Rudolf Virchow. Who completed the cell theory? So Rudolf Virchow completed the cell theory. What Rudolf Virchow has said? So he said that cell arise from pre-existing cell. So this is all about cell theory. Now if we see cell, then cell can be of different shape and size. Cell can be of different shape and size. Now here if I say prokaryotic cell, and here it is bacteria. So this is the size, shape and size of a bacteria. As you can see, cocci means it is spherical. Then bacilli means rod shape. Then you have vibrio. It is called comma shape. And finally spirilla means it is spiral. So if I ask you how many types of cells, shape of cells are present in prokaryote, so you will say there are 4 types of shape. The first one is called coccus, then bacilli, then spirilla, and then is the comma shaped, which is called vibrio shaped, right? So remember these 4 shapes. Usually questions are asked which of the following will be rod shape. So it is rod shape is bacillus, comma shape is vibrio, spherical is the cocci. Now if there is only one spherical bacteria, then it is called coccus. If it is two is called di, so diplococcus 2. Then if it is a chain like this, then it is called streptococcus. And finally if it is 3-dimensional, then it is called staphylococcus. Understood? Single coccus, 2 diplococcus, chain, then it is streptococcus, and if it is 3-dimensional, then it is staphylococcus. Rod shape bacillus, 2 bacillus, diplococcus. Clear? Then vibrio, comma shaped, then it is spirillum. Many times you will say 2 types of shape is together. Cocci bacillus means it is neither completely rod shape nor completely spherical, or it is appearing spherical also and rod shape also. Then it is called cocci bacillus. But remember, these are the 4 main shape given in NCERT, so we will go with that. Some variations are there, like some of the bacteria can even appear filamentous, right, as we have seen in cyanobacteria, filamentous. So now let's move ahead. This is about the size of the cell. Now overall if we see the life, so these are atom. Atom is the smallest, and as we are moving, the size is increasing. So as we can see, atom, then these are molecules, protein, virus, then bacteria. Mitochondria and bacteria are similar in shape. Then you have plant cell, animal cell, then human egg, frog egg, ostrich egg. As you can see, it is the largest cell. Which is the largest cell? Ostrich egg is the largest cell. And so if I ask you to arrange them in correct order, and if I give you, so atom is the smallest, ostrich egg is the largest. So this is the correct order. Now we will see them one by one. As the size will increase, you can see it through the naked eye. So if I ask you till animal cell you can easily, and plant cell you can easily see it through your eye. Then smaller than that you can see through the light microscope. And finally the smallest one you can see it through the electron microscope. So there are three ways you can see. First, directly by your eye. Second, using the simple microscope. And third is by using electron microscope. So if I ask you this question, virus can be seen through, so virus can be seen through electron microscope, protein can be seen through electron microscope, lipid, atom, all of them can be seen through electron microscope, but bacteria, plant cell, mitochondria, animal cell, all of them can be seen through light microscope. And chicken egg, ostrich egg, these all can be directly seen through the eye. They all are example of single cell. So this is about a comparison of how different cells are there in terms of size. Now further if we go and see, so earlier this was the size of prokaryotes and now this is the size of eukaryote, shape of eukaryote. So if we see eukaryotic cell, this is RBC. RBC is biconcave in shape. This is WBC, it is amoeboid in shape. This is pillar shaped, which is long and narrow. This one if you see, it is branched and long. This one is elongated. This is elongated and it has tapering ends, means narrow ends. And this one is round and oval. Now question will be asked, what is the shape of RBC? Then you will have to say shape of RBC is biconcave. What is the shape of WBC? Amoeboid. Elongated shape, this is trachid. Round and oval, so you will say mesophyll cell. Branched, you will say nerve cell. And elongated, so you will say pillar cell. Clear? Now here let's see some of the sizes of different. So if I say virus, so virus the size is 0.05 to 0.1 micrometer. Mycoplasma, we have already seen, it is the smallest cell. It is the smallest cell. What is the size? It is 0.3 micrometer as per NCERT. What is the size of bacteria? So it can range from 1 to 10 micrometer. But if we see NCERT, there they have given 1 to 3 micrometer also and 1 to 5 micrometer also. So you have to remember this. Then yeast is the eukaryotic cell. So eukaryotic cell are comparatively larger, up to 10. Then here some other yeast, although it is eukaryotic, but it is smaller in size and it can be up to, and then finally some other cells which can be longer. So here I will write you a summary. If question is asked, what is the size of RBC? What is the size of bacteria? What is the size of eukaryotes? Right? Now, so these you have to keep it in mind. Now, if I say largest cell, what you will say? Ostrich egg. If I ask smallest cell, mycoplasma. Size of mycoplasma, 0.3 micrometer. Size of prokaryotic cell, it is 1 to 10 micrometer. So this till here everything is clear. Now, so on this basis, on the basis of shape and size, I hope all of you are paying attention here. So let's see some question. Let's see how much you have retained and how much you can answer. So if you are ready for question, give me a thumbs up. Let's start with a question. All of you, no other chatting. Just keep your hand away from chat. When you see question, start replying with the answer. So here we go with the first question. So this is the first question. The shape of bacteria Streptococcus is, yes, Divya, correct. Pinky, okay, just now answer. Reshma, Pooja, all of you are ready for the question. Yes, now answer. Let me see who answered the question first. The shape of bacteria Streptococcus is. Streptococcus. See, the name itself is clear in the term. See, Streptococcus. Coccus means cocci, right? Coccus means cocci, and cocci means it is spherical. So, answer for the first question will be shape of Streptococcus bacteria is coccus. Then, next question, second. So, this is a very simple question. Now, here Vibrio means comma shaped. Comma shaped. Now, this one, the bacterial shape that is spiral and twisted is called, again very simple, spiral. So, spiral will be it, which is a spiral. Okay, Pinky has given answer 2C. Spiral. Okay, Gayatri C. Mohit C. Khalil C. Venkata C. Tanish C. Very good. So it is C. Spirillum. Right? Correct. Spirillum is the right answer here. Very simple question. Now next. Which of the following is largest bacterial shape? Which is largest? Which is the largest shape? We have seen all the shape, but which one is the largest? The largest among them is, so here the coccus is the largest one. Remember, coccus is the largest in shape. Then size of most of the bacteria typically range from. What is the typical bacteria size range from? Now here, understand nanometer and micrometer. Here two options are given. Micrometer is also there and nanometer is also there. Right? So size we have seen in terms of micrometer. So don't confuse with the nanometer. Now will it be A or B? Will it be A or B? Fourth. Correct answer will be, so fourth, the correct answer will be 10. We have already seen that the size of bacteria is up to 10 micrometer. The smallest bacteria is PPLO. All of you note it down. PPLO is 0.1 micrometer. It is a type of Mycoplasma. Mycoplasma is also a bacteria whose size is 0.3 micrometer, and normal bacteria can be between 1 to 10 micrometer. So can we say bacteria range from 0.1 to 10 micrometer? Yes, it is correct. So first is the correct answer. Very good. So this is about the shape of bacteria. Last question from the shape. The bacterial shape Vibrio referred to. Vibrio is what kind of shape? Vibrio, 5. Vibrio, what is the correct answer? Khalil has given answer as C. 5C. Yes, Prasanna C. Tanish C. Khalil. Okay. Fine. So all of you are answering 5C. So correct that Vibrio is the comma-shaped bacteria. Vibrio is a comma-shaped bacteria. Correct? So shape-wise everything is clear. Everything is clear. Now next. So in cell we have seen two types of cell. We have seen two types of cell. One is called prokaryotic cell and eukaryotic cell. Prokaryotic cell and eukaryotic cell. So before we move ahead, all of you, about more than 200 students are connected. So I expect all of you like this video. All of you like this video, and then let's proceed. So I can see 25 have already liked the video. Rest I am seeing how many of you are liking it. So quickly. Now let's see prokaryotic and eukaryotic cell. So these are the two types of cell, prokaryotic and eukaryotic cell. So overall, if you see the comparison between prokaryotic and eukaryotic, both of cells are different in their complexity. Prokaryotic cell are simple cell with few cell organelles, but eukaryotic cell is a complex cell with large number of cell organelles. You all must have studied about the difference between prokaryotic and eukaryotic cell, right? So there are several differences. We will see them. So some difference we will see here. The first difference is in the nucleus. First difference is in the nucleus. Now here what we say, prokaryotic cell, they lack true nucleus. They lack true nucleus. Right? True nucleus means nucleus here is without cell nuclear membrane. That's why it is called nucleoid. Nucleoid is without nuclear membrane. So that is the first difference, because the nucleus is without nuclear membrane. The second difference is that here the cell organelles are membrane bound. Here cell organelles are not membrane bound. So they have membrane bound cell organelle absent. Membrane bound cell organelle absent. Right? Here the membrane bound cell organelles are present. So if membrane bound cell organelle are absent here, so can we say that no compartmentalization? See, if there is a large room, and if I build wall in it, then it will be several compartment. So wall is just like the membrane. If there is membrane, then compartmentalization is there. No membrane means single large room. So no compartment. So compartmentalization is present in Compartmentalization is present in eukaryotic cell, but it is absent in prokaryotic cell. Clear? It is absent in prokaryotic cell. One by one we will see. These were the basic difference. There are many other. We will see gradually one by one. So let us start with first the prokaryotic cell. Let's start with the prokaryotic cell. So this is a typical prokaryotic cell, as you can see here. Now this prokaryotic cell, this part, see here, this part is showing the nucleoid. Just observe all the part of this prokaryotic cell. So this is the nucleoid. Then you can see this is the flagella. This is the flagella which help in movement. Then outer covering, capsule, cell wall and membrane, together they are called cell envelope. So they have outermost cell envelope. Inside is the cytoplasm, right? Cytoplasm is the semi-fluid matrix. Semi-fluid matrix which is present. Then they also have ribosome. Ribosome membrane is present or absent? So in ribosome, ribosome is basically membrane-less. Ribosome is membrane-less. Apart from the circular DNA present in nucleoid, they also have a plasmid, which is also circular DNA. So they have two types of DNA. We will see that later. Along with flagella on the surface, they can also have pili. These are some of the parts of the prokaryotic cell. Now one by one we will see. So let's first see the outermost covering, that is called cell envelope. So we are going to see the cell envelope. It is very important. All of you pay attention, because from cell envelope I have planned to ask a lot of questions, and you will learn a lot of new things here. This you will not have studied till now. So this all things we are going to cover is new. Now all of you who are doing unnecessary chatting, just keep away from the keyboard. No need to type anything until and unless I ask question. Here is the new concept, that is cell envelope. So first of all, cell envelope consists of how many layers? So it consists of 3 layers. 1, 2, 3. So first point, cell envelope has outermost capsule, cell wall and cell membrane, right? This capsule is also sometimes called as glycocalyx. Glycocalyx. So cell envelope consists of capsule, cell wall and cell membrane. So let's first understand about the glycocalyx. So glycocalyx, it is present in some bacteria. It is present in some bacteria, but it is only present in bacteria. It is only present in some bacteria. It is only present in bacteria. Glycocalyx is not present in eukaryotic cell. It is only present in prokaryotic cell. This glycocalyx can be of 2 types. It can be capsule or it can be slime layer. As the name suggests, slime layer is loose and slimy in nature. It will be sticky, whereas capsule is tough, hard and uniform layer. Now this will provide additional protection. So capsule provide additional protection to the cell, additional protection to bacteria, additional protection to bacteria. So now here it is just like a extra protection present on the surface of bacteria. So bacteria which has an extra protection, will it be more dangerous or less dangerous? If it has an extra layer of protection, then when it enters into our body, they can cause more harmful. So harmful bacteria are usually having capsule because they can protect themselves from our immune system. So usually bacteria with capsule are harmful bacteria. Harmful bacteria is also called pathogenic bacteria. Harmful bacteria is called pathogenic bacteria. So remember this point, all of you, that if capsule is present, it provides additional protection to bacteria. That's why it is pathogenic in nature. What is the nature of capsule? So capsule is made up of glycocalyx, right? Now if it is present in a glycocalyx present in a tough, hard and uniform layer, it is called capsule. If it is loose, slimy, sticky, then it is called slimy layer. Correct? Now slime layer helps in attachment. It helps in attachment. So is this point clear? Glycocalyx provide additional layer of protection to bacteria. True or false? If I ask you, glycocalyx provide an additional layer of protection to bacteria. True or false? Answer it in the comment box. Answer it. Answer it. Is it true or false? Glycocalyx provide additional layer of protection. Yes. It provide additional layer of protection. Yes. Reshma telling true. Vicky true. Venkat Ramana, true, very good. Pooja, Lakshya, Vijay, very good. So true. So glycocalyx will provide additional layer of protection. Very good. Now let's go to the second part. We have already seen cell wall of bacteria is made up of peptidoglycan. Peptidoglycan. Cell wall of bacteria is made up of peptidoglycan. So here let's see this in a little more detail about cell wall. So cell wall of bacteria cell wall is made up of peptidoglycan. Now, this peptidoglycan provide, so let's say this is the peptidoglycan of bacteria. This peptidoglycan, if it is thick, then such bacteria will form the gram-positive bacteria. And if this layer is thin, then it is called gram-negative bacteria. Simple. See, bacteria is divided into two parts. Bacteria can be gram-positive, bacteria can be gram-positive, bacteria can be gram-negative. Now question will be there, what is gram here? So gram, actually there is a scientist whose name is Christian Gram. He developed a staining technique. He developed a staining technique which is called gram staining. It is called gram staining, right? Staining is a technique, is very simple. Let's say you have a white t-shirt and you put it into yellow color. Then if you take out your white t-shirt, it will become yellow. So this is called staining. You are staining with a different color. So now staining, after that when you wash your yellow, yellow t-shirt, and if it again become white, means it lose the stain, right? If you wash the t-shirt and if it again become white, means it lose the stain. But if it remain yellow, means it retain the stain, right? So gram positive will retain the stain. It retain the stain, but gram negative will lose the stain. So the one which lose the stain is called gram negative, one which retain the stain is called gram positive. And what is the cause? So the cause is this. The cause is this. If there is a thick peptidoglycan, then it is gram positive. If there is thin peptidoglycan, then it is gram negative, right? So gram positive, gram negative. So understood this concept? This concept is clear. Bacteria is divided into two parts, gram positive and gram negative, based on the nature of cell wall, that is peptidoglycan. If peptidoglycan is thick, it is gram positive. If it is thin, it is gram negative. Gram positive will retain the stain, gram negative will lose the stain. Who gave this gram staining? So it was given by Christian Gram, right? So now, so this is all about the cell wall in bacteria. So cell wall, bacterial cell wall is made up of peptidoglycan. Now, innermost layer of bacteria is the innermost layer of the cell envelope is called the cell membrane. So cell membrane of prokaryote and eukaryotes are similar. We will see cell membrane in the eukaryote in a detail. Just you understand here, cell membrane is more selective in permeability. Cell wall allow entry, but cell membrane do not allow the entry. It is selectively permeable. Clear? So I hope that you all must have understand the cell envelope. And as I said, there is a question from cell envelope. So let's do the question. Are you all ready? Ready for the question? No other message typing. Just tell me if you are ready, then only I will show the question. Are you ready? Yes, Khalil is saying yes. Ready. Very good. Okay. So let's start. Let's start with the question. So this is the first question. The capsule in bacteria helps in. What does the capsule help in? We have just seen, tell me, capsule is the outermost layer, right? See, three layers we have seen. The outermost layer is capsule, middle layer is the cell wall, and innermost layer is the cell membrane. So see here, this is capsule, second is the cell wall, and innermost is the cell membrane. So now tell me, what is the function? What is the function? So 4, correct answer. Answer is coming C. Reshma, Bhaskar, Sheikh, Avnish, Ishwar. Very good. So all of you are giving answer C, right? Venkata, Durga. Very good. So its function is protection. Due to presence of capsule, there is an extra layer of protection. Second question. The outermost layer of bacterial cell envelope is, just now I have said there are 3 layers. Which is the outermost layer? Which is the outermost layer of cell envelope? Outermost layer. Okay, so I am getting, Khalil is telling capsule. Okay, Tanish, C. Pinky, C. Reshma, C. Bhaskar. Very good. So C, outermost layer is the capsule, middle layer is the cell wall, and innermost layer is the cell membrane. Then next, cell wall in bacteria is made up of, just we have seen, cell wall is made up of, what will be the answer? Cell wall is made up of, cell wall is made up of peptidoglycan. We have just seen cell wall is made up of peptidoglycan. On the basis of cell wall, bacteria can be gram positive. So gram positive have thick cell wall and gram negative have thin cell wall. Next, let's go to the next question. A well organized and thick glycocalyx is called. Now this is good question. We have already seen that glycocalyx can be divided into 2 parts, capsule and slime layer. So you have 2 options to select, whether it will be capsule or whether it will be slime layer. Whether it is capsule or whether it is slime layer, well organized and thick glycocalyx. Yes, Tanish is telling C. Divya is telling B, Durga B, Avnish A. Okay, so different answer is coming. B, Reshma B. Okay, now some of you are changing your answer also. Fine. So see, well organized and thick glycocalyx. So well organized and thick glycocalyx is capsule. It is thick and regular. This one is loose, loosely organized. So well organized and thick will be capsule. So third correct answer is B. Very good. Next question. Loose and irregular glycocalyx. Loose and irregular. Again obvious. The correct answer will be B. Slime layer. Slime layer is loose and irregular. Next question. So this is already we have done. Well organized and thick is the capsule. Now the main function of capsule in bacteria is, what will be the answer for this? Main function of capsule in bacteria is, this also we have although done, but it is protection, right? Protection from desiccation and immune system. They enter into our body. So our body will try to kill the bacteria. So killing of bacteria is called desiccation. So bacteria with capsule will be protected against desiccation, and they are also protected against our immune system, right? Now, so then some more cell organelle of prokaryote. So one cell organelle of prokaryote is mesosome, right? So mesosome is what? Let's say this is the cell. So the membrane will fold like this. Folding of membrane, and this is called mesosome. Folding of membrane. As you can see, this is the membrane and this is folded inside like this. This is folded inside and this is called mesosome. Clear? So it is a membrane infolding. It is a membrane infolding. Now, what is present in mesosome? So, mesosome has enzymes. What kind of enzymes are present? They have respiratory enzymes. They have respiratory enzymes. They can be present in the form of vesicle, tubule, right? And they will help in cell division. They will help in cell division and DNA segregation. DNA segregation. So, mesosome, will it increase surface area? Question. Will mesosome increase the surface area? Yes or no? Mesosome increase surface area? Yes or no? Tell me. Mesosome increase the surface area or not? So yes, mesosome increase the surface area. So this is the last topic. Time is already up. So I will complete this and I will stop. So just 1 more minute. So it increase surface area. So now this question is, this term is very much clear. Mesosome increase the surface area. They have respiratory enzyme, help in cell division and DNA segregation. So now question. So last question for the class. All of you answer. Mesosome in bacteria are primary involved in. They are primary involved in. They are primary involved in. So they have, as I have already said, they have respiratory enzymes. So they will be involved in aerobic respiration. They will involve in aerobic respiration. Although to some extent DNA replication they also, but it is mostly the cell division. They will be involved in cell division, right? So, mesosomes will help in aerobic respiration and they will help in cell division. So, we will continue with this chapter in the next class. In the next class, we will be discussing more important and interesting thing, and from the next class, we will start reading NCERT in class also, because I know still many of you must not be reading NCERT. So that habit also we have to develop, right? So all of you must have downloaded the NCERT. So for today still I can see only 74 likes are there. More than 200s are connected. So it is expected that you like the video. Okay. So now we will stop this class. Thank you all of you for the patience, and keep on reading, keep on enjoying. Thank you.